Hello dear students, welcome to Biology Decrypted. Now, after this series of chemistry fundamentals, after going through 4 lectures of chemistry fundamentals, now you are well equipped to understand the concepts of biochemistry. Let's enter into the world of biochemistry through biomolecules. And the first biomolecule that we will be talking about is amino acids. So, in this lecture, that is lecture 5 of Biochemistry Decrypted, we will be talking about amino acid structures. Now, to start with, let's talk about the general structure of an amino acid. So, we will try to draw the general structure of an amino acid. Here on, I'll be writing amino acids, I'll be denoting it as AA. So with this symbol, I'll be denoting amino acids for simplicity. Now, you need to know that this is a wedge and this represents a bond that is out of the plane. That is, if this is the plane, it is coming towards you. And if there's a dash, then it is into the plane. That is, if this is the plane, this is coming towards me. So this is the basic meaning of wedge and dash. We have already referred to it multiple times. If you have difficulty in remembering what is wedge and what is dash, what you need to do is draw something like this. This person is standing above the plane, and this is his shadow is actually going into the plane, and this is how you can remember it, the basic concept of wedge and dash. Why did I tell you? Well, we'll actually draw the structure of carbon, which has 4 bonds. So it is bonded to hydrogen, it is bonded to NH3 plus group, then it has this R group, and it is also bonded to COO minus. It is bonded to COO minus. Now, as you can see here, this particular structure is of a tetrahedron. This is one triangular face, this is another triangular face, this is another triangular face, and this is the 4th triangular face. So overall, as you can see here, this particular structure is actually of a tetrahedron, somewhat like this, and you can actually have the carbon atom sitting here, and these are the 4 bonds, and these are obviously the 4 faces, and therefore it is called as a tetrahedron, because obviously in tetrahedron, tetra means 4 and hedron means triangular faces. So this is the overall concept. Now, if you see here, these are the 4 groups. These are 4 different groups. One is hydrogen, one is NH3 plus, and this group is called as an amino group. Similarly, this group is called as carboxylate group. Now, what is carboxylate? Let's talk about it. See, basically, this particular group, this particular functional group, that is COOH, this is called as carboxylic acid. And now, when you actually, since this is an acid, if you try to ionize this H plus, if you try to remove this H plus, so this will go away as H plus, and what will be left is COO minus. This is basically the conjugate base of a carboxylic acid, which is also called as carboxylate, or simply carboxylate ion. If you try to see how this carboxylate ion looks like, actually it is actually like this. Carbon is bonded to an oxygen, and there is a double bond between the two. Then there is an oxygen. This has lone pairs and there is a negative charge. This is carboxylate ion. This is carboxylate ion, and what happens is when proton is removed, that is when this carboxylic acid is actually deprotonated. I hope you are getting the name. Deprotonated simply means removal of a proton, that is removal of an H plus ion, and you are getting the conjugate base. So this is basically the conjugate base of carboxylic acid. As simple as that. We already know about the concept of conjugate base. Now let's move forward. What is this? This group is NH3 plus. But how did this NH3 plus, where did it come from? So this came from NH2. NH2 is the famous amino group. Now what happened is NH2 took up a proton and therefore it was protonated. Therefore it was protonated, and as a result NH3 plus was formed, and this is amino group. Just like this is your carboxylate group, we can write simply carboxylate group here. Similarly, this is your amino group. And if you look at this particular carbon, this carbon is known as alpha carbon, because the carbon that is directly attached to a functional group, here the functional group, the principal functional group is COO minus. So this is the functional group, and the first carbon that is connected to a functional group is known as the alpha carbon. So these groups can also be termed as alpha amino group and alpha carboxylate group respectively. So both these groups are alpha amino and alpha carboxylate groups respectively. Now what is R? H is obviously hydrogen, but what is R? R actually denotes the side chain. And in every amino acid, actually this alpha amino group and alpha carboxylate group is essentially the same. Hydrogen is also present, but what differs and what actually confers different properties upon them is their side chain, that is the R group, and this R group determines the nature of the amino acid. So what is important is to know that this carbon, in case of your alpha amino acid, this carbon is obviously sp3 hybridized. This carbon is sp3 hybridized. It has a tetrahedral structure. And therefore the bond angle will obviously be 109 degree 28 minutes or simply 109.5 degree. This is very important to note. Now let's move ahead. Let's talk more about the basic concept of ammonia and amines. So let's talk about the basic concept of ammonia and amines. This is very important to know because we will be talking about amino group, amino group, etc. So we should know the basic concept of ammonia and different amines. So amines are basically derivatives of ammonia. So you can say that the derivatives of ammonia are amines. Now, if we try to draw the structure of ammonia, this is how the structure will be. There will be 3 hydrogens bonded to nitrogen, and nitrogen will have a lone pair. And this is the structure of ammonia. Now, we'll be drawing the structure of a primary amine. What we'll do is, if we actually substitute one hydrogen, if one hydrogen is substituted with a R group, then it is known as primary. Amine. If 2 hydrogens are replaced by R groups respectively, if there are 2 R groups, then in this case this will be called as a secondary amine. And if we actually replace all these three groups. If we replace all these three groups with the different R groups, what we will get is a tertiary amine. And now finally, what happens is if we already remove all these three H groups, we replace it with R, and then finally this lone pair is given to another R group, and then finally this gets attached to it. So what we see here is nitrogen is actually attached to 4 different R groups, and therefore nitrogen eventually carries a positive charge. So this is an example of quaternary amine. So there are different types of amine possible. Let's talk about each of them one by one. Now, talking about primary amine, what you do here simply is you will be replacing one hydrogen by a R group, and here you will be having hydrogen respectively. So this is your primary amine. This is your primary amine. As simple as that. Now moving forward, so this can also be denoted as RNH2. This is simply NH3. This can be represented as RNH2. Now moving forward, we will be talking about secondary amines. Secondary amine. This group is also called as amino group. This is also called as amino group. And let me ask you a question here. So your question is, which amino acid has an amino group instead of an alpha amino group? So this is your question for today. Do give the answers in the comment section below. I'll be waiting for your answers. So moving ahead, if we talk about secondary amines and obviously about the amino group, so what we'll see here is this is how the structure looks like. If there is one R group, there is another R group, that is R dash. If there is another R group, that is R dash, and the other one is obviously H. So this is how secondary amine looks like. Now let's talk about tertiary amine. Let's talk about tertiary amine. This is tertiary. Now, as I told you earlier, there will be three different R groups: R, R dash, and R double dash. If they are considered to be different, they may be equal as well. So this is an example of tertiary amine. Now let's talk about quaternary amine. That is, this is your quaternary amine. Now if you're talking about quaternary amine, nitrogen is actually bonded to 4 different R groups: R, R dash, R double dash, and R triple dash. This is how 4 different R groups are attached. Nitrogen has an overall positive charge. And what is the best example of compound where you find this kind of a quaternary amine in biological systems? Well, let me remind you. Have you heard about choline? If not, you'll be learning about it in our class of lipids. Whenever we'll be discussing lipids, I'll be telling you about choline in phosphatidylcholine, but let me tell you right now. So this is how choline actually looks like: CH3, CH3, CH3. Nitrogen has a positive charge, and then this nitrogen is actually bonded to CH2, CH2, and OH. This is choline, and this is an example of quaternary amine. This is an example of quaternary amine. So this is choline, and this is an example of quaternary amine. We have already seen ammonia and all the amines that it can form. Now let's talk about proton. Let's talk about protonation, protons in general. So we'll be talking about protons. What is proton? Well, proton is H plus. But why is H plus called as a proton? See, what happens is hydrogen has one electron. It has one electron, and if this one electron is also lost, what remains inside the nucleus is nothing but a proton. So obviously, if there is hydrogen atom, with a proton without an electron, it can be simply called as a proton because it is not constituted of anything else. It only has a proton and therefore it itself is called as a proton. H plus is itself called as a proton. So this is something very important. Now if we talk about COOH group, this is COOH group. Now what can happen simply here is this H plus will actually go away because this oxygen hydrogen bond is already electronegative. Oxygen will try to pull the electrons towards itself. This hydrogen will be actually released as H plus ions, and what will remain is COO minus. This is your carboxylate. This is your carboxylate, and this process of removal of H plus ions is called as deprotonation. This process is called as deprotonation. Similarly, if NH2 comes here and it actually takes up a proton and it forms NH3 plus. If it forms NH3 plus, then this is called protonation since it is taking up protons. So this process is called protonation. Also, the reverse can happen. NH3 plus can also get deprotonated, or it can undergo deprotonation. Just a moment. So yes, it can undergo deprotonation to form NH2. This is an important concept. Now let's move forward. Let's talk about the general structure of different amino acids. The general structure of almost all amino acids, I would say all other amino acids. Why all other amino acids? Because we'll keep one amino acid separate. Why? For obvious reasons, I'll tell you. What happens is we keep proline separate here. Proline is separate because proline has a secondary amino group. It lacks a primary amino group. So a primary amino group is not present in case of proline. Rather, what is present is a secondary amino group. A secondary amino group is present, and this secondary amino group is also called as an amino group. This is also called as an amino group, and the structure looks like this. There is a carbon here, then there is this hydrogen, there is this COO minus. Then what happens is this carbon backbone is there. There is another carbon here. Then this carbon comes here. Then it goes here, and finally it actually gets bonded to the nitrogen. It gets bonded to the nitrogen, and what we see here is there are two bonds. There are two bonds formed here by nitrogen. There are two bonds, so these two actually behave as R groups. These two actually behave as R groups. So what we can say is this is one R group. This is one R group, and this is another R group. So there are two R groups which are present here, and therefore we can say that this nitrogen is actually not a primary. This is not a primary amino group. This is actually involved in a secondary amino group or an amino group. So this will obviously have a positive charge with a hydrogen bonded to it. This is how we have the structure of proline. We'll be talking about each of the structures one by one, but as of now you should remember that it is fundamentally different from all other amino acids because all other amino acids have a primary amino group and not a secondary amino group, whereas proline has a secondary amino group which is also called as an amino group. So talking about the structure, this is how the general structure looks like. We have already talked about it once. This is how NH3 plus group is present here, and obviously this is a primary amino group. And if you see here, a hydrogen is attached here, then carboxylate group is present here. This is your alpha carboxylate group, alpha carboxylate. And similarly, this is your alpha amino group. This is your alpha amino group. And obviously this is the alpha carbon. Now to this we have attached the R group. To this carbon, the R group is attached. And R group is obviously the side chain. R group is the side chain. Now, this side chain, if this side chain is not hydrogen, let me tell you very clearly, if this R group is not hydrogen, this is a very important condition. In that case, if it is not hydrogen, then we will say that all four groups are different, which is very obvious. We can say that all four groups are different if R is not equal to H. And here the central carbon, in this case, since it is actually having sp3 hybridization, there is tetrahedral arrangement, tetrahedral geometry is there. What we can say here, if all the four groups are different and if R is not equal to H, in that case, the central carbon atom will actually be called as chiral. This will be called as a chiral carbon. This alpha carbon will be called as a chiral carbon, and chiral carbons are generally represented in this manner. This is how they are represented. So this is a chiral carbon. But what if R is H? In that case, there will be H and H, two same groups, and if two groups are same, then we cannot say that all four groups are different. And if four groups are not different, in that case, the central carbon atom will not be called as chiral, rather it will be called as achiral. And we have an example of achiral amino acid. Well, the achiral amino acid is called glycine. So see, we have glycine. Now, glycine is the simplest possible amino acid. So here it has a hydrogen, it has NH3 plus, then it has another H and COO minus. This is the basic structure of glycine. There is this alpha carbon, and we can say that this particular carbon does not have four different groups, and therefore it is achiral. The molecule itself, the whole glycine molecule itself, is achiral in nature, and therefore it does not show any kind of optical activity. So any kind of optical activity is not shown by glycine, whereas by other amino acids optical activity is definitely shown because they have four different groups and they will be showing optical isomerism. So if we take a mirror image for glycine, this is how somewhat the mirror image will look like. Here we have the carbon, here there is hydrogen, here there is COO minus, here there is hydrogen, and here we have NH3 plus. Now, what does chirality mean? See, my hands are chiral. How can I say this, that my hands are chiral? Well, if I don't lift it, up the plane, if I don't move it beyond the plane, like if I am not actually moving it, I'm actually keeping it in the plane. Like this is the plane which you are looking at. Now, if I am just trying to bring it close, this is the mirror image, because this hand and this hand, they are looking like mirror images. Now what I'll do is I'll try to bring this particular hand here. What do you see? They are not superimposing. There is no definite overlap. And this is an example of chirality. This is an example of chirality. Without, even if you rotate it on the plane, you cannot superimpose it. You cannot superimpose it if you are not lifting it from the plane. If you are lifting it, then it's a different story. But that you are not allowed to do that. So keeping it on this plane itself, this will never superimpose. This will never superimpose. But here what you can see is four groups are different. Well, in that case, is not chiral, it is rather achiral, and there is no such non-superimposable relationship. We don't talk about enantiomers in this case. But if you take an example of a chiral amino acid, let's say alanine. Alanine is another simple amino acid. What we can do here is we can draw the structure of alanine. Here we have the alpha carbon, then here we have NH3 plus, we have H, then we have COO minus, and we have, just a moment, we have COO minus and we have CH3 group. So this is the R group here. Obviously it makes all the four groups different, and therefore this particular carbon is chiral in nature. Now if we try to draw the mirror image, this is how the mirror image would roughly look like. CH, here we have COO minus, here we have NH3 plus, and here we have CH3. This is the mirror image. And what we see here is both these mirror images, since this is chiral in nature. So here, these are examples of non-superimposable mirror images. These are examples of non-superimposable mirror images, and they are called as enantiomers. So obviously they are non-superimposable like this because the molecules are chiral. So what we see is they are called as enantiomers. And these enantiomeric forms can have different absolute configurations. So if we talk about absolute configuration, they can have two different forms. Either they can have a capital D form or they can have a capital L form. So these are the forms, these are absolute configurations that are arbitrarily assigned. Now, you might have heard about small d and small l. Small d and small l. Actually, these two signify optical activity. These two signify optical activity, where D, small d, actually stands for dextrorotatory, and it actually represents plus. Levorotatory denotes minus. So the optical activity, whenever it is dextrorotatory, so it actually rotates the plane polarized light in a clockwise direction, or it is also represented by plus. That is, the values will be in plus. So that is an example of dextrorotatory. Now if we talk about levorotatory, obviously it will be rotating plane polarized light in anti-clockwise direction. So this is about clockwise, and this rotates plane polarized light in anti-clockwise direction. This is very important to note. Now let's talk about D and L forms. See, if we take an example of a reference compound, so this reference compound is a 3-carbon aldose sugar or a monosaccharide. This is glyceraldehyde. What we are seeing here is Fisher projection structures, and this is how Emil Fisher gave representations of three-dimensional molecules. He represented three-dimensional structures on a two-dimensional surface. So this is his way of denoting these structures. So these are called as Fisher projections. Whatever I have drawn here, these are the Fisher projections given by Emil Fisher. Now, as you can see here, this is the structure of glyceraldehyde. Obviously, there will be a CHO group, and there is this OH here, there is OH here. This has 3 carbons. So this is glyceraldehyde. Now, what was arbitrarily assigned is based on the position of this OH group. Based on the position of this OH group, what was determined is if this OH is present towards left, then this will be the L form. And if OH is present towards right, this will be denoted as capital D. And since then, this has been followed. This particular notion has been followed, and this is how this again, this is very important to note that this has nothing to do with the optical activity. Optical activity is it is denoted by small d and small l. This is capital D and capital L, and this talks about the absolute configuration and not about the optical activity of different compounds. So if we talk about the absolute configurations, so whenever we talk about D and L forms, we are actually representing the absolute configuration. Now, the absolute configuration in case of this one here, it will be L. So this will be called as L glyceraldehyde, and here this is D-glyceraldehyde because of the position of OH. Since OH is towards left, it is called as L-glyceraldehyde, and if it is towards right, then it is called as D-glyceraldehyde. This is very important. These are mirror images, and these are non-superimposable mirror images of each other, and L and D forms are obviously, they are enantiomers. Obviously, these two are enantiomers as well. As you can well identify this one, this is actually alanine. This is alanine, and this is alanine as well. But these are two different configurations, and configurations that are mirror images of each other. So by comparing the structure to the reference compound, obviously here your reference is glyceraldehyde. So glyceraldehyde here is acting as a reference, and if we talk about the presence of alpha amino group, if this alpha amino group is present towards left, then this will be called as L-alanine. This will be called as L-alanine. If this amino group, this alpha amino group, is present towards right, in that case it will be called as D-alanine. This is just in comparison to D-glyceraldehyde and L-glyceraldehyde. There we talked about the OH group. Here we are talking about the alpha amino group. And this is how we get L and D forms. This is very important to know. Now, this is actually based on the specification of a particular carbon's configuration. So this is with respect to a particular single reference carbon. Now, this is obviously for amino acids, it is the alpha carbon. We are actually denoting this configuration with respect to the alpha carbon. Now, all amino acids have these two configurations except for glycine, because glycine won't have mirror images. So obviously the glycine won't have this L and D form. So what I had actually shown here, there won't be two different forms. You can try to draw a mirror image, but again, this is essentially the same thing. There won't be a separate compound. This mirror image would not be a separate compound. Rather, it will be the same compound because it has two groups that are same. This is what I wanted to point out. Please note this. This won't exist. This will be the only form that would be existing, and it won't have D and L forms. So we don't talk about D-glycine or L-glycine. So it's simply glycine. Now let's move forward. And now we'll be talking about D amino acids. Well, let me tell you that in nature, the most abundant type of amino acids is obviously L amino acids. L amino acids are found almost everywhere. These are actually incorporated by ribosomes. So ribosomes actually incorporate these amino acids. Now, what about D amino acids? There are D amino acids in nature as well. Well, D amino acids are found in peptidoglycan. D amino acids are found in peptidoglycan. And if you talk about peptidoglycan, there is this glycan part, there is N-acetyl glucosamine, then there is this linkage, glycosidic linkage, then there is N-acetyl muramic acid, and this is how these are bonded. To this N-acetyl muramic acid, we have a tetrapeptide. We have a tetrapeptide, or in some cases we have pentapeptides. If you talk about E. coli, for example, there you actually find, in this tetrapeptide, you find D-alanine, then you find D-glutamate. This is how you actually find different types of D amino acids. Well, L amino acids are generally found, but D amino acids, even though rare, but they are found in biological systems. Now let's move forward. Let me tell you one more important thing. We are discussing about alpha amino acids here, right? So this is your alpha carbon. This is your carboxylate group, alpha carboxylate group. This is your alpha amino group. This is hydrogen, and this is your R group. This is your R group. And this is basically, as I told you, this is Fischer projection. This type of representation is known as Fischer projection of alpha amino acids. This is a Fischer projection. Now, how there is one more important thing. If there is a long R chain and if it has multiple carbons, actually, how will we denote it? The answer lies in chemistry again. This is how we'll write COH, we'll write carbon, 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 carbon. This is how we can keep on writing carbons. Now, if we try to number it from IUPAC, like following IUPAC system, this will be 1, 2, 3, 4, 5, 6. We'll always start with this functional group. Since this is the part of a chain, we'll be starting the numbering from carboxylic carbon itself. Now, this carbon, as you know, this carbon which is directly attached to the functional group. So the carbon directly attached to functional group will be called as the alpha carbon. Next, if you talk about Greek alphabets, it's alpha, beta, gamma, delta, epsilon, and so on. So this is the way of writing. So just like we write A, B, C, D, E, there they have alpha, beta, gamma, delta, epsilon. And so if you actually write it like this, this will be alpha, beta, gamma, delta, epsilon. So these carbons are denoted by these Greek alphabets, that is alpha, beta, gamma, delta, epsilon, based on the distance from the functional group. So this is how we denote these. Now let's start talking directly about the structures of all the 20 amino acids. And not only the 20 amino acids, we'll be talking about all the 22 amino acids. So, well, if we want to classify amino acids, if we want to classify all the 20 amino acids, these will be classified based on their R groups, obviously. So there are 20 amino acids, well, the standard ones. Apart from this, there are two more amino acids. And these two amino acids are selenocysteine, and this is your 21st amino acid. And there is pyrrolysine. This is your 22nd amino acid. Very important to note both these amino acids. So there are total 22 amino acids, out of which 20 are the standard ones, and these are the modified amino acids, that is selenocysteine and pyrrolysine. Now let's talk about the nature of the R group. First, we'll talk about these 20 amino acids. Our discussion will be mainly based on the structures of 20 different amino acids, and finally in the end we'll be talking about these two amino acid, and we'll talk about the structures only. In this particular class, we'll be just talking about the structures. So based on the nature of the R group, based on nature of R group. As you know, the general structure has R group. Based on the nature, we can actually categorize amino acids into these categories. So we can have non-polar amino acids. So if the nature of the R group is non-polar, then we'll have non-polar amino acids. Similarly, the R group can be polar but uncharged. Yes, the bond has polarity, but overall it is uncharged. It is not having a charge, to be specific. Now, there can be charged amino acids as well. These can be the categories. Also, if we try to denote a classification differently, well, we can have a classification of all aromatic amino acids. Now, I will be writing certain one-letter codes. These are very important. From now onwards, we will be denoting amino acids by their one-letter codes. We'll also learn about three-letter codes. We'll learn about one-letter codes. And this is how from now on, throughout your course of biochemistry, throughout the course of cell biology, whenever we'll be talking about amino acids, I will be denoting them either by their three-letter codes or by their one-letter code. So it's your responsibility to learn all these three-letter codes and one-letter codes in this particular lecture itself, because it won't be repeated again. You'll have to comprehend all the amino acids just by their one-letter codes or three-letter codes. So keep focusing. Now, the non-polar amino acids can be written as Gavli, Gavli, and then we can write Monday, Wednesday, Friday, and P. Gavli, MWFP. This is a simple mnemonic of writing all the non-polar amino acids. If we talk about the aromatic amino acids, it's simply WIFE, W, Y, and F. I'll tell you about the full names and the three-letter codes. Don't worry. Let's proceed. If I talk about the polar uncharged amino acids, it is ST and CYNQ. ST sync. And if I talk about the charged amino acids, so amino acids can have either a positive charge or they can have a negative charge. So if they have a positive charge, these amino acids are called as basic amino acids. Similarly, if they have a negative charge, they are called as acidic amino acids. These will be known as acidic amino acids. Now, what I'll tell you is how to remember this. You will just say RKH, that is Rak, and here we'll say they. So we'll say Rak they. For basic amino acids, it's RKH, and for acidic amino acids, it is D and E. So now if we actually see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, and 20. All the 20 amino acids, I have actually written them according to their one-letter codes. And here we have actually repeated it. So this is another type of classification. This is just the categorization of aromatic amino acids. So out of these 20, W, Y, and F, these are classified as aromatic amino acids. But if you talk about the main classification, this is the main classification given here. This is the main classification which I have given here. Non-polar, polar uncharged, charged, and in charged we have basic and acidic amino acids. Now, let's talk about the structures of all these amino acids one by one. Now, let's recall the general structure of amino acids again. General structure of amino acid. Well, I'm talking about all the 19 amino acids except for proline, because proline is an example of amino acid, and it's not a true amino acid because the group is a secondary amine group, that is an amino group and not an amino group. So this is something that should be noted. So the general structure of amino acids, we'll be considering this kind of a general structure. This is hydrogen and this will be the variable R group. This will be variable, and it can either be, as I told you, it can either be non-polar, or it can be polar uncharged, or it can be charged. And also if it is charged, it can be basic or it can be acidic. So we'll talk about each of these amino acids. Let me tell you, this is glycine, this is alanine, this is valine, this is leucine, isoleucine, this is methionine, this is tryptophan, this is phenylalanine, this is proline, the important one. Now there is, in case of polar uncharged, these were all the non-polar ones. Now in case of polar uncharged, we have serine, we have threonine, we have cysteine, we have tyrosine, we have asparagine, and we have glutamine. Then in case of charged, in case of basic amino acids, we have arginine, we have lysine, we have histidine, and for acidic amino acids, there is aspartate and there is glutamate. This is aspartate, this is glutamate. Now let's talk about all of these. We will be talking about the R group. So what I have done is, for all the upcoming amino acids, I have kept this particular structure constant, except for proline. For proline, it will be slightly different. It will be NH2 in this case, and there will be bonding here. So there will be two R groups. That is just in case of proline. Otherwise, this particular structure, I have kept this particular part constant for all. So this is something you should draw. This is how you should draw. It's better if you actually take a chart paper, if you take a big chart paper and you start drawing all these structures one by one. Even if you draw just the R group, that would suffice, that is sufficient for remembering all the structures, because this is the part that is variable. This part is remaining the same in case of all the 19 amino acids except for proline. Now we'll talk about all the amino acids in one shot. One by one, all the 20 amino acids. Get ready for the action. Let's start. We will talk about glycine. How to draw the structure of glycine? It's very simple. The group here, the R group here is H. The R group here is simply H. It is hydrogen. So the R group is H here, and glycine. For glycine, the 3-letter code is GLY or gly, and it is the 1-letter code is G. It is very simple, as simple as that. There is nothing complicated here. The group here, R group here, is H. Now moving on. Now as you know, glycine will be achiral. Glycine is obviously achiral because there are two same groups. You can see H here and H here. Now moving forward, we'll come to alanine. In case of alanine, this group is CH3. So we'll write simply CH3 here, right? So this is the R group, and obviously for alanine, now let me tell you one simple thing before we move on. I will be representing, strictly, I will be representing carbons as these kinds of structures. I'll be showing carbon like this, as simple as that. This is how I'll be showing carbon. This is an example of methylene group. This is how we show methylene groups, that is bond CH2 bond. This is the way of representing methylene groups. And now if I actually talk about a methyl group, then I'll draw it like this. This is how I'll represent a CH3 group. So from now on, if I draw these green colored balls, don't get confused. Remember that I am actually trying to denote carbons. And whenever there are carbons, obviously to satisfy the valency of carbon, there will be hydrogens as well. So either I can make a methylene group or I can make a methyl group. It's as simple as that. Let's move forward, because from here I'll be drawing these ball-like structures. So here I'll simply draw this one ball. I'll draw this ball, and I hope you can understand this is nothing but CH3. This is CH3. So in case of alanine, the R group is CH3. And now let's talk about the 1-letter code and the 3-letter code. If I talk about glycine, for glycine it was GLY. For alanine, it is the first 3 letters. We'll be writing alanine as ALA, and the 1-letter code is A. Simple. Let's move forward. Here it was methyl group. The R group was methyl group. Now we'll come to valine. In case of valine, actually there is a CH group, there is a CH3 group, and there is a CS3 group. Now, let me tell you about some important groups again before we move forward. This is very important to note. You should know these groups before you move forward. See, this kind of a group, this group is called as N-propyl group. This is N-propyl group. This is N-propyl group. Now, if we move forward, this is an example of isopropyl group. These are all carbons. Let me tell you, these are all carbons. So this is isopropyl group. Isopropyl group. Now, this is about 3 carbons. If there are 4 carbons, I'm talking about substituents here. This is simply methyl. This is ethyl. So this is ethyl. This is methyl. I've already told you about methylene. This was methylene. Now, these are the 3 carbon groups. Now, if you talk about 4 carbon groups, either it can be in a straight chain like this. This will be called as, this group will be called as N-butyl. Then we can have this kind of a group where this will be called as, this is the secondary carbon and this is also the second carbon, so we'll call it as sec-butyl. We'll call this as sec-butyl. Then there is another possibility. We can have, after N-butyl, we can have sec-butyl. After sec-butyl, we can also have isobutyl. Now, let's make a logo of Mercedes like this. Or you can say you can make a Y like this. Now, we have to make 2 Ys here. There are 2 possibilities. For example, if there is isobutyl group, there can be isobutyl group and there can be tertiary butyl group. Isobutyl or tertiary butyl. If we have isobutyl group, actually the carbon which will get attached is this one. It is this kind of, like, these boundary carbons are being involved. But if we talk about tertiary butyl group, actually this carbon will be attached. This is very important to note. This is the fundamental difference between isobutyl, tertiary butyl, sec-butyl, N-butyl, isopropyl, N-propyl, etc. There is a reason why I told you this. Actually, this isopropyl group is actually found in valine. Sec-butyl group is actually found in case of isoleucine, and isobutyl group is actually found in case of leucine. So this was the purpose behind telling you about this concept of chemistry, about substituents. Now, if we talk about alanine, this we already know it. If we talk about valine, what we'll do, we'll actually draw an isobutyl group. What we'll draw here is a ball like this, and this. What you can see here is a V-like structure. This is an inverted V-like structure which is directly attached to the alpha carbon. So this is another way in which you can remember this. Obviously, this particular group is your isopropyl group. Let me name it. This group is isopropyl group, and your amino acid is obviously valine. We'll take the first 3 letters here. The 3-letter code is val and the 1-letter code is V. And you can just see a V-like structure here. There are 3 carbons involved and we have an isopropyl group here. Moving forward, let's talk about leucine. Now, in case of leucine, as we had already seen, we are having a Y that is attached, and that is the corners, that is the edges of the Y-like structure that is attached to this particular carbon. What we'll do here is simply we'll attach this. Here we'll have this Y-like structure. And this group, as you now know, it is also known as the isobutyl group. This is isobutyl group. So we can just say that this is the particular group that is being attached to this carbon, to the alpha carbon. This is a Y-like structure, and this particular group is known as isobutyl group, and this amino acid is obviously leucine, and leucine, the first three letters can be represented as the three-letter code, that is LEU, and the one-letter code is L. So now we have talked till leucine. Now we will be discussing the structure of isoleucine. Now isoleucine, again, we know what the R group is. It is basically a sec-butyl group. So we have 4 carbons. The second carbon is attached like this, and we have the other 4 carbons like this. So we'll draw one carbon here, another carbon, another carbon, and the 4th carbon. This is how we'll actually show a sec-butyl group here, and we'll highlight it. This is a C-like structure. This is a C-like structure which is present in case of isoleucine, and this group is known as sec-butyl group. This is known as a sec-butyl group, and this is isoleucine. This is an isomer of leucine. You can simply call it as an isomer of leucine. And what you can observe here is this particular carbon. See, obviously there is one chiral carbon. There is one chiral carbon in all amino acids, but this is an additional chiral center. If you actually look closely, this whole thing will behave as a group. This will behave as a group. This will behave as a group, and there will be one hydrogen that will be behaving as a group. So there are 4 different groups. This one, and this one. There are 4 different groups, and therefore there is an additional chiral center. Let me write it for you. This is the presence of an additional chiral center. Very important, very important. So for isoleucine, what we'll do is we'll take 3 different letters. This is I, and this is L, this is E. So we'll take L and E from leucine. We'll take I from isoleucine, the first letter, and finally we'll write ILE. This is the 3-letter code for isoleucine, and it is simply I. I is the 1-letter code. I is the 1-letter code. This is how we saw the structure of, we drew the structure of isoleucine. Now we'll talk about methionine. How do we draw the structure of methionine? See, what we'll do here is we'll simply make an S here. We'll simply make an S here. What we'll do is, this is how we'll draw one ball, another one. We'll leave this ball. Then we'll make another ball. Well, this is a S-like structure. This is a S-like structure where there are 3 carbons here. Actually, here you will make a sulfur. Here you will make a sulfur. So here we'll be making a sulfur. This is the structure of methionine. This is as simple as that. Well, if you try to identify what type of a bond is this, first let's highlight it. This is the R group, this S-like structure, and there is an S inside this S. And also, if we talk about this particular bond, this represents a thioether bond. Actually, if you talk about single bond, O single bond, this represents an ether. Similarly, if it is S instead of O, this will be called as a thioether bond. So here we'll be writing, this is a thioether linkage. This particular linkage is an example of thioether linkage. I'll be highlighting it with red. This is your thioether linkage. Now let's move forward. Let's talk about it. This particular amino acid is known as methionine, and as you can see, this thio means sulfur. So now moving forward, this also has the first 3 letters as the 3-letter code, MET, methionine, and if we write the 1-letter code for it, this will be simply M. Now, methionine is done. Moving forward to tryptophan. Tryptophan has a very important structure. Let me tell you about it. Now, here there will be a benzene ring. To it, we'll have a 5-membered ring attached. This 5-membered ring is of pyrrole. Very important. This is of pyrrole. So as you can see, this part is the complete benzene part, and this is fused to this ring. This is benzene, and this part is actually pyrrole. So when you actually fuse these two structures, what you get is indole. Here you get indole ring, and this is very important. This is the structure of indole. Now, indole ring will be present here. But there will be a carbon here. First, there will be a carbon, and to this particular carbon, we'll have indole ring attached. How will it be attached? Actually, from this particular end. Very important. Your indole ring will be attached from this particular end. Now we'll do the numbering. Of indole. This is also very important. Let's just number all these. Yes. Now we'll start with the numbering. How do we number it? This is, if we start from here, from this particular nitrogen, we'll be starting. This is 1. Then we'll move in this way, in this direction. This is 2. This is the third position to which actually it is attached. So this is 1, 2, 3. Then we won't number these bridge carbons. These two won't be numbered. Then we'll come here 4, 5, 6, and 7. This is the way of numbering indole ring. So how will we draw it? We'll simply draw it like this. We'll have this 5-membered ring like this, NH. And then we'll have this 6-membered ring, that is the benzene ring, like this. We'll have the benzene ring like this, this, and this. This is how we draw simply the structure of indole. It is attached via the third position. This is the second, first, then this is 4th, 5th, 6th, and 7th. These are the carbon numbers, and this is the structure of tryptophan. This is the R group. And as you can see, this is a very bulky kind of a chain. This is an aromatic chain. And so you should remember that this is an aromatic amino acid. Apart from being non-polar, this is also an example of aromatic amino acid. Very important. And this particular group is called as indole. And as you can see, I'll try to give you a hint. This is where you can see a W-like structure. If you look at the base carefully, this is a W-like structure, and this is the 1-letter code. This is one way of remembering it. So what you can say here is the structure of tryptophan. This is the structure of tryptophan. If you write tryptophan, well, you'll have to highlight these 3 letters. Not Y, it's not TRY, it's actually TRP. So it's TRP. Now, TRP won't have a 1-letter code of T or R or P. Those are already reserved. So next we'll move to W. So W is the 1-letter code for tryptophan. Please remember this. This is peculiar. This is not very usual, but yes, you have to remember this. Tryptophan has a 1-letter code of W. Now, we'll move forward. We'll come to phenylalanine. Now, this is very simple. Well, you know alanine. This is alanine. Alanine is having a phenyl ring, that is a benzene ring. Whenever benzene ring is present as a substituent, this is called as phenyl. So it is phenylalanine. Now if you try to draw the outline like this, this is also an aromatic amino acid. A bulky chain is present here. So let me write it here. This is also an example of aromatic amino acid. This is aromatic, at the same time it is non-polar. So This is phenylalanine, and you get the sound of F from here. Although it's phenylalanine, but you pronounce it as phenyl, so F. So yes, we don't write P here, we write F here. So yes, based on the sound, we say it's F. So let's write phenylalanine. This is peculiar. The 3-letter code is very simple, it's PHE, but the 1-letter code is actually F. The 1-letter code is actually F. Now let's move forward. Let's come to proline. Proline has a very peculiar structure because again, it has a secondary amino group, or what we commonly call as an amino group. So it is an example of alpha amino acid. It is an example of alpha amino acid. Well, now let's come to the structure. It's very, very, very simple. What you'll have to do is simply make a ball like this, then a ball like this, and then a ball like this, and you just join it with nitrogen. This ring, this particular ring is actually a pyrrolidine ring. Let me tell you about it. This is pyrrolidine. You had seen pyrrole, right? Pyrrole had these 2 double bonds. Here 2 double bonds are not present. This is an example of pyrrolidine ring. And this is present, this is seen in the structure of proline. In case of proline, it is actually a part of the backbone. What you see here is this is actually connected back to the backbone like this. So this is the structure of proline. For proline, it's very simple. For proline, we simply write proline, and we'll take the first 3 letters. The first 3 letters are PRO, this is the 3-letter code, and P is simply the 1-letter code. This is the structure. It's not very peculiar. Well, this is how you actually draw it. So this is obviously, this gives you, this is your secondary amino group. This is actually your secondary amino group, this one. Very important to note, this is not a typical primary amino acid. This is a secondary amino acid or an amino acid. Now let's come to serine. Now we'll talk about polar uncharged amino acids. So there will be some kind of a polar group like oxygen, hydrogen, or sulfur, hydrogen, like this, wherever there will be an electronegativity difference and there will be polarity in the bond. Now let's talk about polar uncharged serine. In case of serine, the group that is attached is a hydroxyl group, as simple as that. There is a hydroxyl group. Well, this is not directly attached. Well, there is this carbon, there is this alanine kind of a structure. Now here there is OH group, there is this OH group, and this OH group will obviously be called as hydroxyl, right? This is hydroxyl group. This is simply your hydroxyl group. And yes, so for serine, which is a polar uncharged amino acid, we'll simply write its name, then we consider its first 3 letters, it's SER. So the 3-letter code for serine is SER, and the 1-letter code is S. This is how we draw the structure for serine, as simple as that. We won't directly attach OH, we'll actually make a carbon and then we'll attach OH. Now coming to threonine. Well, for threonine, what you have to do is you will have to draw a T first. So here you will try to draw a T like this. You'll make a carbon, then you will draw a T like this. You will draw a carbon here. You will write OH here. You will write H here. This is a carbon. This is OH. This is H. And this is a T. This is the structure of threonine. Very important to remember. So what you will do here is, first you will draw this carbon. Then you will draw this T. You will draw a carbon here. You will draw hydrogen here. And you will make OH here. You will draw OH here. So yes, this also has OH. You can skip this H. You can skip this H if you want. This is not very important to draw. You can actually skip drawing this hydrogen. But yes, this will actually be very easy to remember if you draw the hydrogen. If you draw the hydrogen like this, I'll show it with a darker tone so that this is not very prominent. But yes, you can see this T here. This makes it very easy to remember. So this is the structure of threonine. This is the structure of threonine, and this is one more point to remember. If you see, this is one group, this is one group, this is one group, and this is another group, and therefore this particular carbon, this is the beta carbon. If you see that 4 different groups are attached here, this is one group. So this is another additional chiral center. So as you saw in case of isoleucine had more than one chiral center. Similarly, this also has an additional chiral center. This also has an additional chiral center. This is how you actually draw it. This is the structure of threonine. Now if you write threonine, well, it's very simple. You will take the first 3 letters, you will write THR, and you will write capital T. These are the 3-letter and 1-letter codes respectively. Now let's move forward. Let's talk about cysteine. Well, for cysteine, how will you draw it? It's very simple. It's just like serine. What you have to do is you will have to replace this oxygen. Just try to replace this oxygen by sulfur. What you'll have to do is you'll have to draw a carbon, and then there is SH. This group is known as sulfhydryl group, or it is known as a thiol group. Just like OH was called as hydroxyl because there was hydrogen and oxygen, this was called as hydroxyl. This will be called as sulfhydryl. I hope you got the concept. This is how it will be called as sulfhydryl. So let's draw it quickly. This is the carbon. To it we have S and H attached. This S is basically because of this it is called as a thiol group. Thiol, or we can call it as a sulfhydryl group. So this is thiol or sulfhydryl group. Very, very important. Cysteine is very, very important, because what can happen is this kind of a SH group, 2 SH groups can come close, obviously, from 2 different cysteines. There is one cysteine here, there is another cysteine here. This is how these 2 can come close, and hydrogen from here can be removed. Hydrogen can be removed in the form of H2, and obviously we know that the removal of hydrogen is called as oxidation. So this can get oxidized, and this can form a bond known as a disulfide bond. This particular bond is known as disulfide bond or disulfide linkage, and this is very important. If it is formed between 2 cysteines, they will actually result in a dimer formation. This is basically a dimer, and this is known as cysteine. Just look at the spelling. You don't have E here. Just compare these two. You write cysteine, but here you just write cysteine. You don't write E here. It's very important. This is a dimer, but yes, it can be formed if there is a protein like this and you have an amino acid. If you have cysteine here, if you have SH group here, similarly you can have SH group here, a disulfide bond can be formed here and here. So there can be intermolecular disulfide bonds, there can be intramolecular, intrachain disulfide bonds, there can be interchain disulfide bonds. These kind of disulfide bonds can be seen. These are examples of important covalent bonds. If you look at the tertiary or quaternary structures, these are very important. So yes, cysteines make it possible. This is very important. Now for cysteine, again it's very simple. For cysteine, we just take the first 3 letters. It's CYS, 3-letter code, and C is the 1-letter code. Moving forward, let's talk about tyrosine. Well, if you actually hydroxylate, if you add a hydroxyl group to phenylalanine, if you take the example of phenylalanine and if you hydroxylate it, what you will get here, so what you will draw here is phenylalanine, and if you actually add a hydroxyl group here, what you will get is tyrosine. As simple as that. And if you have noticed this is also an example of aromatic amino acid because it has a benzene ring here. So yes, this is also an example of aromatic amino acid. This is also an example of aromatic amino acid, and this is tyrosine. This is tyrosine. So how would you write tyrosine? You'll take the first three letters, and this will be the three-letter code TYR. And now this will be different. Already threonine has occupied T. Now what will you do? You will shift to the next letter, that is Y. You will shift to the next letter, that is Y, and this is your one-letter code. Again, this is a bit peculiar. Remember this. Now let's move to asparagine and glutamine. But before going to asparagine and glutamine, I'll tell you something important. Let's move to the charged amino acids, that is the acidic amino acids. We'll talk about aspartate, glutamate, and then we'll talk about asparagine and glutamine. Because what the case is, let me tell you. Actually, asparagine is related to aspartate. It's related to aspartate because aspartate has this structure is essentially the same. Now what happens here? Here you have one carbon, here you have one carbon, and you have COO minus. Now what you have to do here is, if you compare this aspartate with asparagine, the basic structure is same. Actually, this will be replaced by this COO minus. This is actually an acidic group. If you have CONH2, then this will be automatically called as asparagine. This will be called as asparagine. So let me draw the structure. If there is a carbon here and there is CONH2 here, if there is CONH2 here, obviously this is an amide group. This is an amide group. So this is amide of aspartate. It's amide of aspartate. It is called as asparagine. Now, if you actually write the name of asparagine, well, the letters you will take is A, S, and N. So this is A, S, N. And for this, the one-letter code that you will write is N. Very, very important. Very, very important. It is peculiar. Now, moving to glutamine. Again, you will have glutamate. Now, for glutamate, if you draw the structure, again, this part will be the same, COO minus, NS3 plus, H. Now, what you'll have to change here, is this particular part. There will be one carbon, there will be another carbon, and here you will be writing COO minus. Now, if you try to replace this in an exact similar manner, if you try to replace this with CONH2, well, you will get glutamine. So how will you draw it? You will draw this carbon here. Now you will add another carbon, and here you will write CONH2. Again, obviously this is also an amide. This is also an amide group, and this is the structure of glutamine. As simple as that. Well, if we talk about the name, glutamine. For glutamine as well, you will take GL and N. So you'll write GLN, and for GLN, this is this is the three-letter code, and the one-letter code will be Q. For GLN, it is Q. Now we have already seen the structure of aspartate and glutamate. Let's just make it once again. This is the structure of aspartate. This is the structure of aspartate. And let me tell you that this is beta carbon. So this particular group can be called as this group. Let me tell you, it can be called as a beta carboxylate. This can be called as a beta carboxylate group. So yes, this is beta carboxylate. Similarly, if we talk about glutamate, there will be two carbons, and then there will be COO minus. So what we can call this as is a gamma carboxylate, because obviously the carbons here, this one is your alpha carbon, this is your beta carbon, this is your gamma carbon, and to it we have a carboxylate group attached. So we can call it as a gamma carboxylate group. So yes, this is gamma carboxylate. So this is beta carboxylate, this is gamma carboxylate, let's just write the names again. Let's just write the names. So we'll write aspartate. And obviously, if there would have been COH here, we would have written aspartic acid. So this is the salt of the acid. This is the conjugate base of the acid simply. So for aspartate, we'll take the first three letters. It is ASP. Three-letter code is ASP. But there is a problem here. It's ASPD. Because in chemistry, you talk about SPD block, right? So after SP, there comes D. So D is the one-letter code. Obviously, it's peculiar. It's not very obvious from the name. So yes, you have to remember this. Now moving forward, for glutamate. If we talk about glutamate, for glutamate, it will be the first three letters, GLU, and, well, let me tell you one important thing. Whatever information I have told you, you'll have to stick it. You have to stick it inside your brain, like inside the walls of your brain with a glue. Well, GLUE. This is the trick to remember it. So yes, you have to actually stick all these information with a glue, and this is your glue. Glutamate, GLU and E. Again, it's not very obvious from the name, but this is how you have to remember it. So this is the one-letter code and three-letter code for glutamate. Also for aspartate, you've seen it. Now let's come to the last part, that is your basic amino acids. We'll talk about arginine, we'll talk about lysine, and we'll talk about histidine one by one. Now, let's talk about arginine. Let's talk about arginine. Well, for arginine, what you have to do is, this is a very nice trick. You have to walk 3 steps. What do you have to do? 3 steps. Step number 1, step number 2, step number 3. And then you get inside your Mercedes. This is your Mercedes, right? This is the Mercedes logo. And you'll have nitrogen here, you'll have nitrogen here, you'll have nitrogen here. Here you'll have a double bond. And you'll have to satisfy all the valencies of nitrogen, as simple as that. This is your Mercedes. What you will do is you will actually paste the Mercedes logo here. You'll actually paste the Mercedes logo here. You will be attaching this N here. Then there is this. You have this double bond. There is this nitrogen. There is this another nitrogen. And I'll try to actually make a circular logo here for simplicity. Yes, this is how I'll draw it. And here we'll try to satisfy the remaining valencies of nitrogen with hydrogen. Obviously, there will be NH2 here. There will be NH with a positive charge here. Obviously, there will be a positive charge since this is a basic amino acid. There will be positive charge here, and there will be another hydrogen here. This is the structure of arginine. This is the structure of arginine. And what I have drawn here, this particular group is a famous group. It has a very high it has a very high basicity. Actually, it is very highly basic. So this group is called as guanidino group. This group is known as guanidino group. This is the simple structure of arginine, where you walk 3 steps and you actually get inside your Mercedes. Next, we'll talk about lysine. Well, for lysine, it's even more simple. Now what you have to do is you have to walk 1 step, 2 step, you'll have to walk 4 steps basically. 1, 2, 3, 4. 1, 2, 3, 4. Now, after walking 1, 2, 3, 4 steps, what you see here is this is your beta carbon, this is gamma, this is delta, and this is epsilon. Now you have reached your epsilon carbon. To this epsilon carbon, you actually have your NH3 plus group attached. To this particular carbon, you have your NH3 plus group attached, and therefore this group is known as, this particular group is known as epsilon amino group. This is known as epsilon amino group. Very simple. Epsilon amino group, or it is sometimes also called as butyl ammonium group. Yes, obviously we can call it butyl because there are 4 carbons involved. So we can call it as a butyl ammonium group or a butyl ammonium side chain. So some books also refer to it as butyl ammonium. Now let's talk about histidine. Let's talk about histidine, then we'll talk about the names. See, for histidine, you have to remember this particular structure. There is a shield like this. there is a shield like this. This is the shield. This is how we have this particular shield in positive. This is how we have the structure. This is the structure. This is the shield we are having, and obviously this nitrogen will have a positive charge. This is a shield like structure. Now what you have to do is simply, here you will have one carbon and here you will have the shield. What this group is, do you know what this group is called as? This is imidazole ring. This is your famous imidazole ring, and in case of imidazole ring, what we do is we start the numbering from here. This is 1, this is 2, this is 3, this is 4, and this is 5. So the fifth carbon is attached to this. The fifth carbon is attached to this. So what do you have to do? You'll simply have to write this. So this is your carbon. Now after this carbon comes this particular structure. You will make this shield like structure like this. Here you will have the nitrogen, here you will have the nitrogen with positive charge, and here you have this base of the shield, and this is how you will draw the bonds. This is your imidazole ring. This is your imidazole ring, and this is your final basic amino acid. So we'll write the names one by one. Well, if we talk about arginine, the first three letters give you the 3-letter code ARG. But now you cannot write A, you cannot write G, you cannot write A, you cannot write G. The only option you have is to write R. The main sound that comes from arginine is arginine. So it's R. Moving forward, if you talk about lysine, well, lysine, the first 3 letters, they give you the 3-letter code, that is LYS. Again, this is peculiar for arginine. For lysine, it won't be L. It won't be L, it won't be Y, it won't be S. Everything is taken. So what you have is the next, like the neighbor of L, that is K. Yes. So it's the neighbor of L, that is K. And this is the only option you had. So for lysine, the 1-letter code is K. And this is also a bit peculiar. Moving forward, if we talk about histidine, now this is very plain and simple here. If we talk about histidine, for histidine is the first 3 letters HIS, and the 1-letter code is H. The 1-letter code is H. So these are all the 20 amino acids, and you can actually see all the 20 amino acids. We have drawn the structures of each of these, and now I hope you know how to draw these amino acid structures. In most of the cases, what you do is you actually try to take a chart, or you just stick a chart and you don't actually remember the structures. Here I have taught you to draw these structures even without seeing. I hope you'll be able to draw these structures from now on. And this is very important. This is very important to understand any kind of aspect of biology wherever amino acids are involved. So let's talk about the 21st and 22nd amino acids respectively. Now we'll talk about selenocysteine and we'll talk about pyrrolysine. So if we talk about the structures of selenocysteine and pyrrolysine respectively, the basic structure will be the same. Here we'll have CO minus, here we'll have NH3 plus, here we'll have H. Now here what we'll do is we'll have a CH2 group, and here, just like serine, we'll have selenium here. We won't have just like cysteine had SH. Here we have selenium and hydrogen. So this is called selenocysteine. Now if we try to draw the structure of pyrrolysine, well, we'll try to draw the structure of lysine first. This is CO minus, this is H, this is NH3 plus. And here, if we try to draw the structure, obviously we'll try to draw these carbons. This is your alpha, beta, gamma, delta, and epsilon. So you have carbons here. Now what will happen is you will draw lysine, you will draw this N of lysine, and what you will do is here you will form an amide bond. This is an example of amide bond, this one. And here you will be attaching this group. Here you will be attaching this group where you have a nitrogen, you have this double bond, and you have another carbon here. So this carbon can be shown as a CH3 group. So this is an example of pyrrolysine. This is the structure of pyrrolysine, and this is your amino acid number 21, and this is amino acid number 22. So these are the two more amino acids, the 22nd and 21st amino acids. Now there is a question at the end. If you have watched this video till the end, I have a question for you. Like, I have multiple questions for you. Tell me in the comment sections. I'll write the question here. What is question 1? 3-letter codes for selenocysteine. Just a moment. Selenocysteine and pyrrolysine. Then there is the second question. What are the 1-letter codes for both? What are the 1-letter codes? And let me tell you, these are actually encoded by stop codons. What are the stop codons each for pyrrolysine and selenocysteine? Well, I'll be waiting for your comments. If you fail to answer, I'll be answering it in the comments as well. So this is all for amino acid structures. I hope now you'll be confident enough to draw any amino acid structures if given to you. Now you'll be able to visualize peptides in a different way, because you are empowered with the knowledge of structures. Now you can draw these yourself with your own hands without even looking at the charts. And this is what I actually wanted. This is how I wanted to empower you with this capability of drawing any particular amino acid structure. So I hope you enjoyed learning drawing structures of amino acids in this video. So in the upcoming videos, we'll be talking more about amino acids. So thank you for watching. Do share this video, do like this, and also comment down in the comment sections. Do give the answers for these questions. See you in the next video. Thank you for watching.